uh, movement. So we earlier on said that the bacteria do reproduce by uh, binary fission, and this is basically where the bacteria just divides into two. Okay, and so the two become daughter cells that are actually independent. So what happens is that before that division happens, you need to have the uh, circular chromosomes replicating such that the daughter cells actually eventually also get uh, these uh, chromosomes. Okay, so the chromosomes duplicate, and then after duplicating, they separate, and then a new plasma membrane forms around these two cells. These two cells are identical. The meaning of this is that they are similar. This type of division is different from eukaryotic division because you have no speed of formation like they basically have lysosomes. So if you have um, bacteria in a test tube and you want to see how many of them will you have after a certain period of time, and you can do that. Of course, there are different ways of counting the bacterial cell. So this is basically what you have. Remember, we talked about culturing, which is an issue to do with growing the bacteria in a particular medium. Okay, so and then another one that I can talk about now is inoculation. When you add bacteria um, to a medium, which is basically nutrient medium, you would expect them to keep multiplying until the time till when they start to decline. So the graph, if you plot the number of the viable bacterial cells against time, you'll get a graph that looks like what we are seeing here, like that. So you'll have an area, you have time when the increase is not so much, and then you have time when the increase is so exponential, and then you have a time when you don't have an increase or a decline, and then you have a decline phase. So we want to explain what happens at each of these phases and why do we have such situations. So the lag phase, here the bacteria is getting used to the new environment, and they need to synthesize the enzymes that they will need, and also substances that they will need for growth. So they are getting used to basically. Okay, so that is the lag phase. So no serious multiplication is taking place. And then the log phase, they are now used to the place. There is enough food. They have the enzymes that can do the work. Therefore, they can multiply very fast. That's the log phase. So you have them increasing in number. And then you have the decelerating. The growth is slower because the amount of food is actually stationary, is actually declining. And then you move the stationary phase where those that are growing and then those that are dying are actually equal. So you don't have a decrease or a decline. And what causes this is that the food is declining together with the oxygen, but also the toxic products are also there that cause this death. Okay, and then eventually you reach a point where those that are multiplying are very few. So you basically have a declining population. Okay, no food, that is no nutrients. The oxygen is also exhausted. You have a lot of waste, so basically decline. The bacteria will always grow. Yeah, as long as there is food, there is oxygen. and also that when the temperature is appropriate. Remember that we have bacteria that can grow in extremely unusual temperatures. We have those that can grow in low temperatures and those that can grow in high temperatures. And then the availability of moisture is very important, because moisture is very important for their uh, metabolic processes. And then we have accumulation of toxins. Okay, the toxins can actually disrupt their metabolic processes. The toxins can also result into a low pH, which inhibits growth, and then also have a buildup of carbon dioxide that lowers the pH because of formation of the folic acid. So the bacteria have very useful applications. We can use bacteria in degrading harmful products or neutralizing harmful substances. For example, petroleum waste dyes and pesticides. So they basically help in cleaning the environment. We talked about the bacteria in the gut that is used for synthesis of very important vitamins. But also in goats, we have bacteria in their guts that produce enzymes that can be used to digest cellulose. And then we talked about the cyanobacteria that are very important in nitrogen fixation. And then the bacteria that is very important in recycling of nutrients. We talked about that in the nitrogen cycle and also in the carbon cycle. Okay, but also decomposers help in recycling, for example, carbon, phosphorus, and nitrogen. Can be used in production of different food products, yogurt, vinegar, and cocoa. Okay. Bacteria can also be used in fermentation, antibiotic production, and also in production of hormones. The autotrophic bacteria are very important in producing nitrogen. Producing oxygen. And then, of course, bacteria also have a number of negative impacts. They can cause diseases to humans and also to humans and also crops and also animals, and can cause food spoilage. And then the sulfur bacteria can produce sulfuric acid that damages metal pipes. And then also certain bacteria can reduce soil fertility by converting nitrates into atmospheric nitrogen, and this contributes to reduction in soil fertility. Okay, so thank you very much for listening.